In the next keynote speech, we will go on and keep exploring the opportunities and application of AI in pharmaceutical area. Today, we are very pleased to invite the CEO of Insilico Taiwan. Insilico is an international company that grows rapidly recently and has become the most influential AI company. Ladies and gentlemen, please welcome the CEO of Insilico Taiwan, Mr. Artur Kadurin.
Hello, Dajahao. I'm trying to learn Chinese, but of course, at this moment, because I took like 1 month of lessons,
let's give a warm welcome. Woo! That was good enough.
Yeah, thank you. Okay, so I will try to add some interesting thing about the AI in the field of medicine, but not in the general, but more precise and in more deep, and maybe for me, of course, more interesting field is about drug discovery. And of course, I will start with talking about money, about the money. So, you know, for the last few years, we developed very few real drugs in the world. And development of a new drug costs a lot of money, really a lot of money. And for the 2012, the amount of spent money was like 2 billion or something like this for development the single drug. And of course, it's easily to understand why, because when you are trying to develop, you have to start from the scratch. You have to start from the disease. You have to find out what the disease is. Then you are trying to find out what kind of molecule, what kind of treatment you want to provide. And you have to perform preclinical validation tests and then put it to the clinic. And the clinic is very, very expensive because you have to work with humans. And there is different phases of the clinical trials, etc., etc. And on each step, you have very, very small chance that your actual molecule or your drug will pass this step. So if on the input, if you are starting with like 10,000 possible candidates for the specific disease, to the clinic will pass like maybe a few tens of them, or maybe in rare cases it can be like 100 of the molecule. And after 3 phases of the clinical trials, most of them, almost all of them will gone, and only one will pass all the steps. And actually failure rate a few years ago was like 92% for the clinical trials. And of course, it's a very, very interesting and very, very valuable place where you can apply AI. Because if you can predict with more accuracy, even if you can reduce this 92% to 90%, it's a really huge amount of the money. Because this 2% from the 2.5 billion is enough to make a new startup. Okay. So, because I'm from the company doing drug discovery, I will talk about our point of view, our perspective of view in this field, what we are doing, what kind of research we are doing, and what is in the core of this. And we have this small, we call it golden triangle, the triangle of target, disease, and the actual molecule. All of this data are collected or generated. Now we can generate data as well from the clinics, from the laboratory tests, and from the computations, from the modeling system. And in each of part of this triangle, actually the big task itself. You can for example, if you want to work on the new disease, probably you need first the clinical data about this disease, then some kind of laboratory tests, and then you want to develop the idea or hypothesis about what this disease is, what target you should address with your future treatment. And we work with all three of these objects. And we split our pipeline, and I think it's actually like a general pipeline for drug discovery, but we put more AI in each step of this pipeline. And we split it in three different phases. So first one is target identification, when we go in from the disease to the target, to the single protein or maybe single cell type or something like this. And we're doing this with AI as well. And this is a big field itself. You can try to start your company in this field, not for the whole pipeline, but for this part. And the second step is actually molecule design. And in the maybe a few years ago, the molecule design was done by bioscientists, by bioinformaticians, with some experts from the chemistry, from biochemistry, etc. And all that they could do actually to write by their hands, write the structure of the molecule, and then simulate some reactions, simulate the, for example, interactions between molecule and protein, between small molecule and protein, and try to predict will it work or not. But at this moment, because of this, because of growing the AI technology, we actually don't need to write the structure of the molecule by chemists. We can generate the structures, and I will show a few more slides about this as well. And of course, after you came with some molecular leads, with some candidates, you want to predict how it will work in clinic. You will pass some laboratory tests for your specific target, for your specific cell line, but after you get this result, you want to predict what will actually happen with human in clinic. And this is the third part of this pipeline. And I will focus on each part a bit more. So when we are talking about the target identification, in the most cases for our company, for example, we are working with cellular data, and we're trying to understand what's going on in cell lines when you put your treatment, when you put your molecule into it. And one of the way to think about it, you have some kind of signature of your cell line. It can be transcriptomic data or proteomic data or many other kind of data. And but because of technology and because of nature of this data, it can be very noisy. Because of course, in every cases, even with the same disease, the cell lines can produce different number of proteins or different number of can express genes in different ways, etc. So even for the same cell lines, time to time you will come with different results. And to work around this, in this field actually works, many of the scientists works with pathway activations, because this protein, these targets in the cell actually connected, combined in the very difficult, very big graph. And you can think about what's going on, not only looking on the single protein, but looking on the chains, on the pathways in this graph. And we built this iPanda engine, which can understand what pathway activated, which pathway activated when you put the drug in the cell or when cell developed the disease. So you can reduce this noise and get more accurate data, which of course will provide you more accurate results. And based on this technology, yeah, of course, this kind of research are published, and we are publishing a lot of papers. I believe that at this moment we published like 70 something in a reviewed journal, of course. So based on this technology, you can actually predict the properties, predict the outcome of using the specific molecule. So if you know how the specific molecule works with the specific cell line, you can predict the results for new molecules using AI, using neural networks. And one of our work was about this. We get data from different databases. This UNPD, KeyGG, it's just a name of databases. Actually, doesn't matter. And we scored the natural compounds from these databases and compared them to known and very interesting modern molecules like metformin or rapamycin. And we found that few of these natural compounds actually works in the similar way. So in this case, you don't need to go to the clinical trials because you can use this natural compound right at the moment you found them as supplementary. So after this research, we put on the market our first supplementary product, and we have more in pipelines. Actually, at this moment, it's more than one product. And this company, Life Extension, it's a quite big US company which are working with a lot of patients in America, and they also provide some sales for those of them who make blood tests. So after we put this product on the market, we not only provide our results and our solution for some cases, but we also get the data back. We build this back loop because now, after 2 years of use this product, we can see what actually happens with those who bought our product and did they react as we predicted or not. And you can think about this like about the clinical trials, but you don't need to work with clinic because it's natural. So it's kind of unique data, actually. I don't know if anybody else doing something like this. And yeah, and at this moment we already put 2 of these products on the market. You can find information and the publications, our publication about this, etc. And we have more in our pipelines, and 2 of these is first one, ageless cell, about senescence cells, and the second one is senolytic and metabolic. Okay. But the first short story was about the classical approach. I can say it's the predictive approach. Most of you, actually, I think that most of you are very familiar with this because, of course, the predictive approach was started much more, I don't know, maybe as in the early 90s, after the second winter of AI, there already was a lot of different solutions in different fields. For example, in image recognition, of course, and in voice recognition with predictive approach. But at this moment, there is another very, very big thing which are happening. The generative approach works, and we can use this generative approach as well. So we built another part of our pipeline based on this approach. We can, at this moment, we want not only to predict properties of the molecule, but generate new molecule with predicted with properties you want them to have. And 2 years ago, we submitted the first paper in this field using this generative adversarial network to generate new molecular structures. And we've done this research on the cancer cell lines. And we get, I'm not sure, the results isn't here, but we get quite good results. The almost half of the predicted molecules from this system, which we could find in the different databases or different libraries of molecular of vendors, was connected to the anti-cancer research. And you see, it took like, at this moment, it took like 6 months to review this paper because it was so innovative. No one did it before. No one did use generative approach for the biochemistry. And of course, this year we are much more advanced in this field, and because of the progress in the generative adversarial network, because of the progress in the AI in general, we can use such words like neural, like adversarial neural computer or something like this. This much more sexier words for the titles of our paper. And of course, this paper is much more advanced, one of our recent papers. So using this pipeline, we can work in the different in the different fields in the same time, because the very big part of our work are done by computers now. Of course, we still have a team of bioinformaticians, of chemists, etc., which providing us their expertise, which are leading this research, etc. But we don't need the thousands of them to work on different diseases, because humans can't calculate all this stuff at this speed that AI provides. So you can see that we started with thousands of the molecules in different, in totally different diseases, and came with some great results in different fields as well. And for the last part, for the third part of our pipeline, when you already have a drug candidate, when you put something in clinics, into the clinic, and for example, you have results from the phase 1, if you can be sure that this particular drug will fail in the phase 2, you can prevent spending a lot of money. If you can be sure that specific molecule will not pass the whole 3 phases or even 4 phases of clinical trials, you can save a lot of money for the pharmaceutical companies or for your company if you are working on this. So even if you can predict with very good probability the results of the clinical trials, because of course it's very complicated, but if you can switch from half to half to say 60% or 65% of accuracy, even in this case, you can save a lot of money for those molecules in which you are really sure. And because it's in the development now, I will not tell you a lot of this, but but we are working on this engine, and probably you will hear about this as well. If you want to look on our publications, you can find us on PubMed or on Google Scholar or something like this. All of these papers are signed by Insilico Medicine, so it's easily to find. And this is just the few of those papers which are, in my opinion, most interesting. But of course, there is a lot of different stuff as well. Okay, so I'm finishing. I'm not sure about question answering session, because I was told that probably after my speech you want to ask something, or you will have the possibility to ask. But looking at the previous speakers, I can see that I'm not sure that it's true. So maybe if you want to ask me about something, about anything, you can find me later here and just ask in person. Of course, I will be walking around for the part of time. Okay, so thank you. Thank you.
Thank you, Artur. Thank you very much.